Deep inside every cell in your body sits a secure biological vault, the nucleus. Inside is the ultimate master recipe book, your DNA. But the machinery that actually builds the proteins, the ribosome, lives entirely outside this vault. The master DNA sequence is too precious to move. Any damage to the original copy would be catastrophic for the cell. To solve this, the cell creates a temporary, disposable copy of a specific genetic instruction. This transcript is RNA, a specialized messenger that can safely carry the code onto the cellular factory floor. DNA is built of two parallel strands. If a cell needed to manufacture proteins at peak speed, simple logic suggests it should copy both sides of the DNA simultaneously to double its output. In the microscopic world, this pursuit of raw speed is a death sentence. Prioritizing manufacturing volume over meticulous precision triggers a sequence of events that shuts the entire cell down. Imagine a cell transcribing both sides of the DNA at once. Two complementary RNA strands emerge and instantly snap together into a rigid, double-stranded cord. This creates structural gridlock. The ribosome is physically unable to grip this fused molecule, causing the protein factory to detach. To prevent this total manufacturing collapse, nature deliberately throttles its own output. It forces the system to ignore half the available information, reading only one strand to ensure the final product remains functional. To keep the process controlled, the cell only copies a tiny, heavily regulated fraction of a single DNA strand at any given time. This precision requires boundaries. A promoter tells the machinery where to begin, while a terminator stops the process. The RNA-building enzyme, polymerase, is locked into a single direction, reading from the 3' to 5' end, ensuring a continuous copy. This directional rule leads to a naming quirk. The strand being read is the template, while the untouched, ignored strand is labeled the coding strand, simply because its sequence matches the final RNA product. These rigid mechanical limits are the evolutionary guardrails that keep the cell's genetic machinery from descending into the chaos of double-stranded gridlock. In simple bacteria, this process is lean and direct. They execute transcription in a single open compartment, using one multi-purpose tool that handles every type of RNA. Complex organisms, like humans, require a massive increase in infrastructure. We utilize 3 specialized enzymes, each restricted to its own line within the cellular nucleus. This added complexity creates a significant mess. The raw RNA copy produced in human cells is heavily polluted with sequences that serve no immediate purpose in building proteins. Increased evolutionary complexity hasn't led to a cleaner genetic process. Instead, it has introduced a dangerous layer of internal data pollution that must be scrubbed before the code is usable. To salvage the raw RNA, the cell initiates an aggressive post-production edit, much like a film director cutting a final reel from hours of messy footage. The useless segments, known as introns, are physically identified and cut out of the sequence entirely. The meaningful blocks of data, called exons, are then stitched back together into a continuous functional script. To protect this edited strand from the harsh environment outside the nucleus, the cell attaches a structural cap and a tail, armoring the ends against degradation. Only after surviving this sequence of cutting, stitching, and armoring is the mRNA considered mature enough to leave the vault and begin building life. Every second, your cells expend massive amounts of energy, cutting away these discarded fragments. It raises a fundamental question. Why would an efficient system preserve so much genetic bloat? The RNA world hypothesis suggests these sequences are ancient molecular fossils. They are remnants of a period billions of years ago when RNA was the primary molecule governing life on Earth. These discarded introns constitute a dormant, historical archive of genetic potential, a library of ancient variations preserved across the eons. By maintaining this deep reservoir of ancient data, the cell keeps a vast library of genetic options on standby. What looks like an inefficient mess of junk today may be the raw material nature needs to trigger the next major leap in human evolution.